Bio 1009, Lab Section 8, and welcome to our PSA. So what exactly sparked our curiosity in using oils to help inhibit the growth of Streptococcus mutans and lactobacilli in the oral microbiome? Well, first we were inspired by coconut oil and the craze surrounding its crazy benefits to the human body. You can see that all over social media, that's for sure. From there, we were able to find articles that supported oil pulling and its benefits to the oral microbiome, oleic acid content in oils, and antimicrobials found in avocados, thus sparking our interest in plant-based oils and their impacts in the oral microbiome. And so our question was, will plant-based oils such as avocado oil, olive oil, and vegetable oil inhibit the growth of Streptococcus mutans and lactobacilli found in the oral microbiome? And from there, we hypothesized that the natural antimicrobials found in the plant-based oils, avocado oil, olive oil, and vegetable oil, will inhibit the growth of Streptococcus mutans and lactobacilli species in vitro.
100 microliters of Streptococcus or lactobacillus was micropipetted onto a plate of agar. The bacteria were spread with an L-shaped spreader. After that, 4 sterile paper discs were placed on the agar with a sterile forceps so that the potential zones of inhibition wouldn't overlap. 10 microliters of each test substance was micropipetted onto 1 paper disc. For each plate, 1 disc was saturated with positive control, 1 with negative control, and 2 with 1 particular oil. The positive control that we used was a diluted toothpaste solution. It was used because it's known to have antimicrobial properties. The negative control is deionized water, chosen because it should have no effect on the bacterial growth. Each week we used each type of oil on both bacterial species, for a total of 6 plates per week. Multiplied by 4 weeks, we used 24 plates in total.
The data that we collected did not support our hypothesis or prediction. We saw no zones of inhibition for any of our oils. However, our positive control was able to consistently produce a significant zone of inhibition, and our negative control did not have a zone of inhibition, which was the expected result. We were not able to use any data collected during the first trial due to contamination, and we were also not able to use any of the data from the 3rd and 4th trials of the lactobacilli plates due to contamination.
Conclusions. None of the oils, avocado, olive, or vegetable, were able to inhibit the growth of lactobacilli or streptococcus. The positive control exhibited a significant zone of inhibition along most trials.
Our possible sources of error are human contamination, such as some of our germs may have made it onto the agar plates. We did not use sterile forceps in the first trial, and we may have not fully sterilized them for the remaining trials. Our olive oil may have been contaminated because it was not from a new bottle. The duration for each trial was not consistent, and we didn't always use the same amount of oils from one trial to the next. And all these errors may have contributed to our results.
Strengths and obstacles. One strength of our experiment was our controls, in that our positive control provided us with a zone of inhibition and our negative control did not. We also kept a very good attitude throughout the entire experiment despite it being a bust. And most of all, we gave it our all. One obstacle that we encountered was that we were unable to do the experiment with wells, so the oil may not have had a chance to diffuse. We also experienced contamination, and our experimental design was changed after the first replication in order to better fit our experiment.